Good day, this is Professor Will, CBMD PhD. Today's Thursday, June 13, 2019. It is 16:12, 13 seconds Eastern Standard Time. And this is a continuation of my scientific research. So let's get started here. Gut microbiomes eat our medication. Date June 13, 2019. Source Harvard University. Summary. Researchers have discovered one of the first concrete examples of how the microbiome can interfere with drugs intended to pass through the body. Focusing on levodopa, the primary treatment for Parkinson's disease, they identified which bacteria of the trillions of species is responsible for degrading the drug and how to stop this microbial interference. Even more crucial are the reactions that happen after the plates are cleaned. When a slice of sourdough travels through the digestive system, the trillions of microbes that live in our gut help the body break down that bread and absorb the nutrients. Since the human body cannot digest certain substances, all important fiber, for example microbes, step up to perform chemistry no human can. But this kind of microbial metabolism can be detrimental. According to researchers, gut microbes can chew up medications too often with hazardous side effects. Maybe the drug is not going to reach its target in the body. Maybe it is going to be toxic all of a sudden. Maybe it is going to be less helpful. Researchers describe one of the first concrete examples of how the microbiome can interfere with the drug's intended path through the body. Focusing on levodopa, L-dopa, the primary treatment for Parkinson's disease, they identified which bacteria of the trillions of species is responsible for degrading the drug and how to stop this microbial interference. Parkinson's disease attacks nerve cells in the brain that produce dopamine, without which the body can suffer tremors, muscle rigidity, and problems with balance and coordination. L-dopa delivers dopamine to the brain to relieve symptoms, but only about 1 to 5% of the drug actually reaches the brain. This number in the drug's efficiency varies widely from patient to patient. Since the introduction of L-dopa in the late 1960s, researchers have known that the body's enzymes, tools, and perform necessary chemistry can break down L-dopa in the gut, preventing the drug from reaching the brain. So the pharmaceutical industry introduced a new drug, carbidopa, to block unwanted L-dopa metabolism. Taken together, the treatment seemed to work. Even so, researchers said there's a lot of metabolism that's unexplained, and it is very variable between people. That variance is a problem. Not only is the drug less effective for some patients, but when L-dopa is transformed into dopamine outside the brain, the compound can cause side effects, including severe gastrointestinal distress and cardiac arrhythmias. If less of the drug reaches the brain, patients are often given more to manage their symptoms, potentially exacerbating these side effects. Researchers suspected microbes might be behind the L-dopa disappearance. Since previous research showed that antibiotics improve a patient's response to L-dopa, scientists speculated that bacteria might be to blame. Still, no one identified which bacteria species might be culpable or how and why they eat the drug. So the research team launched an investigation. The unusual chemistry, L-dopa to dopamine, was their first clue. Two bacterial enzymes can perform this conversion, but a good number bind to tyrosine, an amino acid similar to L-dopa, and one from a food microbe often found in milk and pickles. Lactobacillus brevis can accept both tyrosine and L-dopa. Using the Human Microbiome Project as a reference, research teams hunted through bacterial DNA to identify which gut microbes had genes to encode a similar enzyme. Several fit their criteria, but only one strain, Enterococcus faecalis, E. faecalis, ate all the L-dopa every time. With this discovery, the team provided the first strong evidence connecting E. faecalis and the bacteria enzyme PLP-dependent tyrosine decarboxylase, or TYR-DC, to L-dopa metabolism. And yet, a human enzyme can and does convert L-dopa to dopamine in the gut. The same reaction carbidopa is designed to stop. Then why the team's researchers wondered, does the E. faecalis enzyme escape carbidopa's reach? Even though the human bacterial enzyme performs the exact same chemical reaction, the bacterial one looks just a little different. Researchers speculated that the carbidopa may not be able to penetrate the microbial cells, or the slight structural variance could prevent the drug from interacting with the bacterial enzyme. If true, other host-targeted treatments may be just as ineffective as carbidopa against similar microbial machinations, but the cause may not matter. A research team already discovered a molecule capable of inhibiting the bacterial enzyme. The molecule turns off this unwanted bacterial metabolism without killing the bacteria. It is just targeting a non-essential enzyme. This and similar compounds could provide a starting place for the development of new drugs to improve L-dopa therapy for Parkinson's patients. The team might have stopped there, but instead they pushed further to unravel a second step in the microbial metabolism of L-dopa. After E. faecalis converts the drug into dopamine, a second organism converts dopamine into another compound, meta-tyramine. To find this second organism, researchers left behind his mother's dough microbial masses to experiment with a fecal sample. He subjected this diverse microbial community to the Darwinian game, feeding dopamine to hordes of microbes to see which prospered. Lenta 1. These bacteria consume dopamine-producing metatyramine as a byproduct. This kind of reaction is challenging even for chemists. There is no way to do it on the benchtop. Previously, no enzymes were known that did this exact reaction. The metatyramine byproduct may contribute to some of the noxious L-DOPA side effects. More research needs to be done. But apart from the implications for Parkinson's patients, Lenta's novel chemistry raises more questions. Why would bacteria adapt to use dopamine, which is typically associated with the brain? What else can gut microbes do? And does this chemistry impact our health? All of this suggests that gut microbes may contribute to the dramatic variability that is observed in side effects and efficiency between different patients taking L-DOPA. But this microbial interference may not be limited to L-DOPA and Parkinson's disease. Their study could shepherd additional work to discover exactly who is in our gut, what they can do, and how they can impact our health, for better or worse. Good day, this is Professor Will, CBMD, PhD. Today's Thursday, June 13, 2019. It is 16:56, 48 seconds Eastern Standard Time. And this is a continuation of my scientific research. So let's get started here. Reaching and grasping, learning, fine motor coordination changes the brain. Date, June 12, 2019. Source, University of Basel. Summary. When we train the reaching for and grasping of objects, we also train our brain. In other words, this action brings about changes in the connections of a certain neuronal population in the red nucleus, a region of the midbrain. Researchers have discovered this group of nerve cells in the red nucleus. They have also shown how fine motor tasks promote plastic reorganization of this brain region. Simply grasping a coffee cup needs fine motor coordination with the highest precision. This required performance of the brain is the ability that can also be learned and trained. Researchers have investigated the red nucleus of the region of the midbrain that controls fine motor movement and identified a new population of nerve cells which changes when fine motor coordination is trained. The more that grasping is practiced, the more the connections between the neurons of this group of nerve cells are strengthened. The red nucleus is a little investigated region of the brain. Grasping is a skill that can be trained and improved, even in adults. For muscles to perform a movement correctly, brain commands must be transmitted through the spinal cord. The red nucleus, which over the years has received little attention in the brain research, plays an important role in fine motor coordination. Here the brain learns new fine motor skills for grasping and stores what it has learned. The research team has now investigated the red nucleus in more detail in the mouse model and analyzed its structure and neuronal composition. Researchers have found that this brain region is very heterogeneous and consists of different neuron populations. The research team has characterized one of these neuron populations and demonstrated that learning new grasping movements strengthens the connections between the individual neuron. When learning new fine motor skills, the coordination of this specific movement is optimized and stored in the brain as a code. Thus, we have been able to also demonstrate neuroplasticity in the red nucleus. In a further step, the team now wants to investigate the stability of these strengthened nerve cell connections in the red nucleus and find out to what extent they regress when the learned fine motor movements are not practiced. The findings could also provide new insights into the understanding of Parkinson's disease, in which affected individuals suffer from motor disorder. The researchers hope to find out whether the neuronal connections in the red nucleus have also changed in these patients, and to what extent fine motor training can re-strengthen the neuronal network. Good day, this is Professor Will, CBMD, PhD. Today's Thursday, June 13, 2019. It is 17:18, 18 seconds Eastern Standard Time. This concludes my scientific reading. I hope you enjoyed listening. Have a great day.